I think many young people think, oh, why should I go on and do research? Because all the big stuff's been discovered. No, it hasn't. It really, really hasn't. We don't have much of an understanding of how these receptors are working.
Welcome to the OT podcast, which takes a deep dive into the topics affecting optometry today. Each time we'll find a real expert in their field and attempt to pull everything we can out of their brain so it may reside in yours. This is the 13th episode in the series, and we are recording on the 6th of August 2024. You can listen to the other episodes on the OT website, on Apple or Spotify, or from your usual podcast provider. In our 13th episode, we'll be speaking to Professor Russell Foster. But first, I'll just introduce myself. My name's Kerry Smith-James. I'm the clinical multimedia editor for Optometry Today. As always, with me today,
Ian Beasley, Head of Education for the AOP, clinical editor of Optometry Today and a visiting lecturer at Aston University. Now, Kerry, before we welcome our guest today, perhaps we should just have a bit of a chat about CPD, particularly with end of cycle looming.
Yeah, well, as you say, end of the 3-year GOC CPD cycle. So as always, OT is here to the rescue. We have our survival pack. So as well as the usual OT offering, we're going to have 5 republished CPD articles that you can access as part of your subscription or your AOP membership. And then you also have a clinical interpretation article, which is where you look at images and answer questions about them. And then we've also got a brand new video. This has been done in partnership with Cooper Vision. And this is a video with Nikki Latham. And she's telling us about how to manage the returning myope. So we may now have patients that we've been managing their myopia. But what do you do when they come back for their 6-month or their yearly checkups? And how do we manage them over the future? So that's a really nice video, and that will be 2 interactive CPD points, and as usual there'll be a discussion on the LinkedIn OT Interactive CPD discussion group as well.
So that takes us up to, I think, almost 20 CPD points available in the final stages of the CPD cycle, so that should be enough to help most of our members across the line. But then of course we'll be straight into the 2025 new cycle. And with that in mind, we're already beginning to plan our portfolio of education for next year. Of course, we've got 100% Optical, the AOP's flagship event, which will be taking place 1st to 3rd of March. You can register for that event now. And then bookings will go live early in January, where we'll be able to offer a week of exclusivity for AOP members. And also, CPD in OT will be going live on the 3rd of January. So if you want to get a head start on your points, then you'll be able to do that very early in the cycle.
I think it's a good idea if we may just to remind any of our members that haven't started or haven't completed or uploaded their reflective exercise yet to their MyGOC account, that they need to organise that and do so before the end of the cycle, which is 31st of December 2024.
Thanks, Kerry. So we'll move on and welcome our guest for today. So it's a real pleasure to welcome Russell Foster, who's a Professor of Circadian Neuroscience and Director of the Nuffield Laboratory of Ophthalmology and the Head of the Sleep and Circadian Neuroscience Institute. He's a Senior Fellow at Brasenose College at the University of Oxford, and in 2015 he received a CBE for services to science. Russell's research interests span both visual and circadian neurobiology, with a focus on the mechanisms whereby light regulates circadian rhythms. And he received recognition from around the world for his discovery of a subset of photosensitive retinal ganglion cells. So, Russell, it's a pleasure to have you with us today on the OT podcast. So thank you for giving up some of your time today.
Oh, delighted, Ian. Thank you for asking me.
Kerry and I have been delving into your latest book, Lifetime, and no doubt that will crop up in our conversation later. But perhaps before that, it'd be really nice if you could just maybe go back in time a bit and just outline, your humble beginnings at Bristol and that extended stint in Virginia, and then coming back to the UK up until where you're at now, if that's okay.
Yes, of course. So I went to University of Bristol as an undergraduate to study zoology. And I thought I'd become a marine biologist because I was a good swimmer, competitive swimmer at that stage, and loved biology. So, what do you do? You do marine biology, and then it was in our second year physiology class where we were recording the light responses from an eye of a locust. And I just thought it was so remarkable that you could, you could turn the lights on, different intensities, different wavelengths, and you could see a change in the electrical response you got from the locust eye. And that triggered, an interest in physiology, which I hadn't really had before, because I'd done sort of fossil collecting and, collected insects and all those sorts of childhood things. And then I just got fascinated with physiology. And for my third year undergraduate project, it was recording from the parietal eye of a tadpole. And again, recording the electrical responses. And I was, and in fact, it's an amazing response because if you dim the lights, you trigger a swimming response. So here's a eye photoreceptor that's mediating behavior. And I thought that was just so cool. And we published those data. And that was the springboard for my PhD work, which I did in Bristol with Sir Brian Follett, on trying to understand the photoreceptors that regulate seasonal biology of birds. As the day lengths increase, it triggers the reproductive response in birds. And so you get mating, you get growth of the gonads, you get, nest building, all of those sorts of things. And we had known from the 1930s that it wasn't the eye that was detecting these receptors. It wasn't the pineal. And there are actually light sensing systems within the hypothalamus of birds. And I thought that was so amazing, and we knew nothing about them. And so we worked out some ways of trying to deduce what the mechanisms of what the photopigment was. And so we discovered for the first time that was an opsin vitamin A based photopigment system embedded within the brain of birds. And that was published subsequently in Nature. But that was sort of triggered this whole interest in how photoreceptors interact with timing systems. And then went really after that. I did lots of work in Holland to learn photoreceptor biochemistry, and Germany to do anatomy. And that set the stage for 8 years at the University of Virginia. And I sort of was able to work on reptiles and fish and birds. And one of the key questions we asked at that point was, well, how do mammals regulate their circadian systems? How is the internal circadian day locked onto the external time? Classic mismatch, of course, is jet lag in us. And we knew it was the eyes, because if you cover up the eyes, then the ability to entrain, to set the internal clock to the external world, is gone. But I was puzzled. Again, going back to my undergraduate days, I was lucky enough to have lectures from John Lithgow, who wrote this extraordinary book published in 1979 called The Ecology of Vision. And John's lectures and the book, and I was privileged to work with John actually as part of my PhD work as well, that photoreceptor systems are fine-tuned to the particular sensory demands. So what the visual system does, of course, is grab light in a fraction of a second and then forgets it's seen it. That's how we generate an image of the world. But the clock needs an overall measure of the amount of light around about dawn and dusk. And so it's integrating light information for minutes and sometimes hours. And I suppose I couldn't really understand how the eye, an organ of vision, could do that. So we started working on mice with hereditary retinal disorders. In fact, mice that were being used to study eye disease in humans. And what turned out to be quite extraordinary, and I still remember those early results, was that even though these mice were visually blind, they could regulate their clock absolutely normally. No attenuation of sensitivity whatsoever. When you covered the eyes, the ability was gone. So there seemed to be something else within the eye. And to cut a long story short, over a decade, we generated genetic models whereby all the rods and cones were eliminated and et cetera, and ultimately were able to show in mice that the ganglion cells, those cells of the retina that whose axons project into the brain and form the optic nerve, about 1 out of every 100 of those cells is directly light sensitive. This was also shown by Dennis Dacey in the macaque monkey, and other people were working on rats, for example. We stuck with mice using our rodless, coneless models. And so we were able to show that they are maximally sensitive in the blue part of the spectrum. And so this then gave rise to this sort of fascination of this dual function of the eye. It's the organ of course, space because of vision, but it's also the organ of time because of its ability to regulate the internal clock. And without the eye, then this ability is gone. And of course, much later on, we've studied individuals who have lost their eye. So, for example, working with Blind Veterans UK, and the extraordinary situation where you chat to these individuals and they can deal with vision loss, but they often say, I can't stand being constantly tricked by my body. I feel fooled by my body because, of course, the clock keeps on ticking. But for most of us, it's slightly longer than 24 hours. So these individuals get up later and later and later each day. And one chap told me a story, and these are fiercely independent individuals, saying, I'd lost my eyes, but I was damned if I was going to be the person with the messy front garden. So he'd worked out a way of getting the lawnmower out and, working out how to cut the lawn. And he was in the middle of doing this. And his wife came down and tapped him on the shoulder and said, it's 3:00 in the morning. The neighbors are complaining. And, that gives you, it's kind of funny, but it's actually also tragic because here's somebody wanting to make sure everything is as normal as possible, and yet his body is tricking him. So really it was from a passion of the first recording from those locust eyes through the pineal eye of frogs, then my PhD work, which is on day-length detection in birds, then working on reptiles and fish and mammals at the University of Virginia, that led to this discovery of this third photoreceptor system within the eye of mammals. I should say, we discovered it first in fish by discovering another photopigment gene, which wasn't expressed in the rods and cones, but in the inner part of the retina, which was really very helpful because at the time when we suggested that there's another receptor within the eye of mammals, the response from the vision community was very hostile. I remember in one seminar, a chap stood up and looked at me, and I was waiting sort of for a question. He just looked at me and said, bullshit, and walked out. They couldn't accept the fact that we've been studying the eye for 150 years, seriously, and they'd missed an entire class of light sensing cell. And so to begin with, there was a lot of opposition.
And that same person that yelled out BS, was it the same person that thought that there were some light receptors at the back of the knee, which are...
No, this wasn't the same person. So I came back from the States in 95, and in 1998, a paper came out in Science saying that if you shone light behind the knee, you could regulate the circadian system. Now, we thought we couldn't understand this because if you cover the eyes of humans or any mammal, then the ability to regulate the clock was gone. And but it got huge publicity. And I remember sending a grant off to the MRC, and the reviews came back saying, why is Foster looking for a novel photoreceptor within the eye, whilst we know it's actually behind the knee? And, it was taken very seriously. You still see it in textbooks today, and the paper hasn't been withdrawn. A group at Harvard replicated the methodology exactly and failed to repeat the results. They found no effect. And it looks like there was some technical problems with the study, but it's never been retracted. It sits there. And I'm often still asked questions, in public talks. Oh, what about that receptor behind the knee?
Just picking up on a couple of points that you've referenced already. So you very kindly delivered a talk at the AOP's flagship event, 100% Optical, in 2020. And there you had a focus on the jet lag issue in patients with severe sight loss. And at the time you were, I believe, working on some potential pharmaceutical interventions to help these patients. I just wondered where that work is.
That's a really good question, because it's a good example of where, fundamental curiosity-driven research, how is light regulating the circadian system? And we sort of discover the receptors. And the next question is, well, how do they signal to the molecular clockwork? How do they change gene expression so that you can align this molecular clockwork with the outside world? And so having defined that pathway, we then thought, well, hang on, could we find drugs that will mimic the effects of light on the master clock within the brain? And that's where we got up to. And I think I showed some of our preliminary data. Well, for one of the drugs, we've done all the now preclinical work. And we've actually got, on the basis of the quality of those data, FDA have given us orphan drug status. We then have gone on to show that the drug that we're working on is safe in humans, and we were able to do that with our colleagues in Australia, in Melbourne. So we're now trying to raise the funding for taking this drug to the clinic. The problem is that it's a repurposed drug, which means that it was a drug that was developed for something else. We screened this class of drug for shifting the clock, and it worked. It works really amazingly well. But because and we own the intellectual property for the use of this class of drugs on shifting the clock, but we don't own the drug itself, which means that Big Pharma feels uncomfortable investing. So it's a bit frustrating. We've got something that can, give back a sense of time to the time blind, but we haven't been able to get the funding to do the last bit of the work. So we've got actually some discussions with a group next week, and hopefully we can kickstart this. That's one of the drugs. Other drugs that work on the same pathway do different things. So for example, one of the drugs we've discovered increases the amplitude of the circadian oscillation. And one of the things that's so interesting is that as we age, that circadian drive diminishes. And so that's why we have disrupted sleep and our hormonal profiles. We're getting up in the middle of the night to pee because the hormonal underpinning of urine production is flattened somewhat. And so that's at a much earlier stage, but that's another drug, which is part of that input pathway. In fact, it's really fascinating. It's an epigenetic modifier. It's actually changing the expression of these genes to increase the amplitude of the clock. And there were two other drugs that we're also working on. So progress has been wonderful on the basic science, the preclinical, but it's been slower getting those drugs into clinical practice, which is something that I'm desperate to do before I retire. I mean, if I can give back a sense of time to the time blind, I'll be immensely happy. And having worked with individuals who've lost, their both their sense of space and time, how important, critically important, is to them. So that's my passion and motivation for that.
In the meantime, for these time-blind individuals, is there anything you can do by just having very robust other cues to circadian rhythm? So time when they eat and time when they stop exercise and so on, all those peripheral cues? It's
a very good question. And although I've emphasized the importance of light, which is absolutely the dominant time giver, Zeitgeber, to the clock, you can have an effect with timed eating and timed exercise, but it's really weak. And for most individuals, it doesn't work very effectively. Melatonin, of course, is the hormone produced from the pineal. Melatonin is released at night, and it's a biological marker of the dark, of the night. And some studies have shown that melatonin can have a very weak effect on the clock, but for most cases it doesn't work. In fact, Blind Veterans UK came to us originally because they'd used melatonin, and they said it just doesn't work for our subjects. So have you got anything? And you're and I said, well, we're working on something, but it's not there yet. But that began the collaboration, because melatonin is all that's out there, and it's not very effective. It's often poorly classified as a sleep hormone. It isn't. It's a mild modulator of sleep, and it's a mild modulator of the clock.
You talk about the circadian clock, and we've got to the photoreceptors go somewhere in the brain. And now, could you explain to our listeners, who will mostly be optometrists, they'll have a good grounding in neuroscience, although they won't be neuroscientists as such. Can you explain to us a little bit about how that actually works, the mechanism?
Yes. So, in fact, when I first went over to the States, I worked with Mike Menaker and Martin Ralph on trying to define what area of the brain actually contains the master clock. And as a result of transplant experiments, I mean, these are extraordinary. What Martin found was a mutant hamster. Instead of having a clock of around about 24 hours, its clock was 20 hours, just complete. So it was getting up earlier and earlier and earlier by 4 hours a day. Just extraordinary. And what Martin did, and I helped him, was to destroy the SCN of a 24-hour hamster and then put in the SCN of a 20-hour hamster. And what we could show is that the circadian rhythms were restored with a period of 20 hours. So, the transplanted suprachiasmatic nuclei, that part of the brain that contains the master clock, was able to confer the restored rhythm of 20 hours. So that showed definitively that this small structure within the base of the hypothalamus, the suprachiasmatic nuclei, contained an essential property of the circadian architecture that it conferred, period. And because when you destroy the suprachiasmatic nuclei, you don't see any rhythms, the assumption was that this structure within the hypothalamus was driving rhythmicity on the rest of the body, producing rhythmic hormonal outputs, locomotor rhythms, you name it. And then Uli Schibler, a Swiss scientist, showed that in fact most cells of the body contain a circadian clock. And so we changed the way we viewed the circadian architecture. So instead of this SCN forcing rhythmicity on the rest of the body, every cell in the body seems to have its own capacity to generate a circadian rhythm. And what the master clock is doing is coordinating billions and billions of individual clock cells throughout the organ systems of the body. So you have this extraordinary circadian network. So I've likened it to a bit like an orchestra. You have a conductor of the orchestra, which would be the master clock, the SCN, producing a rhythmic beat from which all the component parts of the orchestra, the body cells, take a reference cue and align their rhythmicity in accordance with the master clock, which is set by the light-dark cycle. So you've got this amazing circadian system. Now, what happens, of course, when you destroy the SCN is that all of the individual clock cells in the body, which are still ticking, but they drift out of alignment with each other. So the apparent rhythm has disappeared. But if we had gone back and measured individual cells, which when we did those early experiments you couldn't do, we'd have seen that we hadn't abolished circadian rhythmicity at all. What we'd abolished was the ability of the whole architecture, the whole system, to work in a coordinated way together. But that's what fooled us for really quite a while. And it was only Uli's work that made us think differently and showed us that, yes, individual cells have a clock. And so, we use the shorthand of the circadian clock, whereas, of course, it's much better to call it the circadian system.
Optometrists have been manipulating light. That's what we do. We take it and we focus it on people's retinas. And we've had a lot of products that filter particular wavelengths of light. So we attenuate blue light, for example, which is, as you've alluded to, is the peak sensitivity of these photosensitive retinal ganglion cells. Should we be messing about with that? Does it have an effect at all?
There's been a lot of discussion about whether blue light can be used positively and negatively. I mean, we sort of got into this because at the time, blue blocking lenses were being used in cataract surgery. So all of these lenses are UV blocking, but they were introduced some blue blocking lenses with a bit more yellow in them to block out more blue light, short wavelength light. And the arguments surfaced that, well, actually what we could be doing is taking away important blue light for the circadian system. And the reason these lenses were developed in the first place is there was some data, which isn't very strong, it still isn't very strong, that blue light can increase age-related macular degeneration and things like that. It's probably good to use a blue blocking lens because of safety, but does it have a downside? And we showed with individuals who had blue blocking versus UV blocking that there was no difference between the two sorts of lenses. And furthermore, the commercially available lenses block less blue light than the lens of a 50-year-old. So actually, they're not a problem. And those observations that we made have now been replicated by many other groups around the world. So there's no problem with blue blocking lenses. And although the data isn't very strong that blue light is increasing age-related macular degeneration and stuff like that, it's probably wise to be cautious. And they don't seem to have a downside. But it's extended to this whole, when we discovered that these were blue sensitive photoreceptors peaking at 480 nanometers, which is sort of the blue of a beautiful blue sky. We might want to discuss why that might be. Is that what about the blue light you get of computer screens and looking at a Kindle and all those sorts of things? And so a group in the States looked at the effect of reading a Kindle. They called it a light-emitting ebook. And individuals looked at this for 4 hours on its brightest intensity setting over 5 days. And they did this just before bed. And after 5 days, it delayed sleep onset by just statistically significantly 9 minutes. And as one of my colleagues at the time said, well, that may be statistically significant, but that's biologically meaningless. Now, it did change the circadian profile of melatonin. Melatonin rhythms were shifted, but it didn't translate into a change in sleep-wake timing, which is a really good point because it shows that melatonin rhythms are not underpinning necessarily the sleep-wake cycle. But some really important data a few years after looked at allowing people to have light exposure for about 6.5 hours during the middle of the day of about 500 lux or so. So still not that bright, but fairly bright, because in the original experiments, people had been kept under dim light prior to the light exposure. And the Scandinavian group that used the sort of light exposure during the day showed that even subtle effects and the melatonin effects of looking at a Kindle before you go to bed were completely abolished. So one's light history is really important in this context. And so, lots of recommendations have come out saying you shouldn't see more than 40 lux per 2 hours before you go to bed. And really, these are based upon laboratory studies, not real-world studies. And it also doesn't take into account the duration of light exposure. So many of the studies that have tried to get an effect of light on the circadian system have used durations of 6.5, sometimes 7 hours. So hugely long stimuli. And what we don't know to make recommendations are, well, the wavelength of light, the duration of light, when you're exposed to light, dawn versus dusk, for example, what light history you've had, how much light you've seen in the day, your angle of gaze, and how old you are. We know that the circadian system gets less sensitive as you age, and that's true for both humans and mice. We did some studies on these a while ago. So whilst I think it's sensible to minimize light exposure before you go to bed, because brighter the light, the increased levels of alertness, and that will delay sleep. It's normal screen use, domestic light setting is probably going to have very little or any effect at all on the circadian system. So I think there's you know how things go. So it's all rush. Oh, blue light is bad. You shouldn't do this. You shouldn't do that. Now it's all swung back the other way, and people are actually saying, well, there's no evidence for these programs which go from a blue enriched to a red enriched screen. Or is there any point in wearing yellow blue filtering glasses? Well, nobody's getting any effect of these devices. So I think we do certainly need more data, but the situation is nowhere near as scary as I think the initial report suggested, and how it's been amplified in the press. I should say also by some scientists who've pushed their data beyond where I think many of us are comfortable. And it's the press office in so many cases that wants to make a story, and so they exaggerate the findings.
With the daytime history of light exposure in mind, should that influence how environments such as care homes are lit and so on? As a domiciliary optometrist, I would go to care homes quite often and have to give patients a gentle prod to wake them up, and the residents around them would all be dozing off at 11 a.m. And
I think this is an incredibly serious issue. First of all, the circadian system becomes less sensitive as you age. And also, if you're inside all the time, you're not getting robust exposure to light. So average office lighting conditions, let's say 300, 400 lux, maybe. If you're lucky, inside a care home, it can be much lower than that. But outside, even on a grey day, you're going to be exposed to 10,000 lux or more. Sunny day, it's going to be up to 80,000, maybe even 100,000 lux. So we live our lives in dim, dark caves, but we've evolved, of course, a system which has been adapted to brighter lighting conditions. That's what we see in the real world. So we're not very sensitive to dim light. And so there's some studies that came out of Holland which increased the amount of light in the day areas of nursing homes to 2,000 lux or so. And the bedrooms were in darkness, and they improved sleep-wake cycles in those individuals by providing a robust light-dark cycle. But also, very importantly, those individuals who were showing low levels of dementia, their cognition improved very significantly after the improved sleep. So the light-dark cycle, stabilizing the circadian system, stabilizing the sleep-wake, then has knock-on effects across, the health spectrum, not least on individual cognition. So light in the nursing home is critically important.
And I think, in addition to that, if you've got someone in a care home with sight loss, if they're amongst a group of people that are chatting away because they're more alert, it might help, serendipitously keep the people with sight loss awake, or help keep their, just because they know stuff is going on, that it helps reinforce the fact that it's daytime and not nighttime.
I think you raised another important point, Ian, which is that you can have significant sight loss. You can lose your vision, but those photosensitive retinal ganglion cells can still be there. So we worked on an individual, and we showed that this person had no conscious light detection at all, and yet she was able to regulate her circadian system. And I should say that those photosensitive retinal ganglion cells are regulating more than just the clock. So we showed early on in our rodless, coneless mice that there was an intact or a partially intact pupil constriction, which of course is really interesting. So they're used as brightness detectors, and they contribute to our pupil constriction. The way we sort of identify this lady is because although she was visually blind, she still showed a slow pupil constriction in response to light. And so we were then able to go on and do sort of further tests on this individual, showing that her circadian system could be entrained. Her pupil constriction was there. It would maintain constriction under bright light, but also some other things. So what was really extraordinary is that Dennis Dacey's work on the macaque had shown that these photosensitive retinal ganglion cells project to the dorsal geniculate nuclei. Well, why would they go to part of the visual relay within the brain? And so we then asked our individual with no conscious light detection, if you were asked to say when the lights were on and off, would you mind just telling us? And she said, no, there's no point in doing this. I am completely unaware of light. I said, yeah, we understand that, but just indulge us. And so Farhan Zahidi did his experiments and showed that she would always know when the lights were on and off. So it looks as though these PRGCs are not only regulating the clock, but they're brightness detectors, regulating pupil constriction, an awareness of light at some level. And they're also projecting to other structures in the brain, perhaps associated with hormone release and indeed emotional structures and alerting structures. So we're just really scratching the surface of this other photoreceptor system. And I should also say that whilst we originally conceived the system as being visual and non-visual, we now know that the rods and the cones can modify the electrical activity of those PRGCs in ways we don't fully understand. But that means, of course, that the spectral range at which the clock is sensitive to could be fairly broad. It's not just 480 blue light, but it could well be getting inputs from the rods and the cones. And some of the models that are being built all sort of assume that the rods and the cones add to the light responses of the PRGCs. But Dacey's work on the macaque, on the monkey, has shown that the short wavelength cones seem to be inhibitory. So there's some real complexity. And another layer of complexity is that we originally thought, well, it's just one melanopsin expressing ganglion cell, which is the photopigment of these extraordinary receptors. But there are at least 5 morphological classes going to different structures in the brain. So it's turning out to be what we originally thought was simple. It's not a simple system at all, as all biology is. It's layer upon layer of complexity. And I think the excitement over the next, 10, 20 years is for the next generation of vision biologists is to work out what they're doing and how they're doing it, which I think is such an exciting question.
Gosh, I mean, that is, as you say, that's what drew me into biology as an exciting field, because just when you think you know it all, there is another layer of complexity to it.
I think many young people think, oh, why should I go on and do research? Because all the big stuff's been discovered. No, it hasn't. It really, really hasn't. We don't have much of an understanding of how these receptors are working. Okay, we know what the cells are. We know what the photopigment is, 480 nanometers, and it's called OPN4 or melanopsin. And I should also unpack the story of melanopsin because Iggy Provencio was my PhD student, and we did a lot of the rodless coneless work together. And anyway, he left the lab to do a postdoc saying, I'm never going to work on mice again. They're smelly, they're expensive, and they bite me. So he went off to work on the skin cells, the melanophores of the African clawed toad. And he was interested in them because if you shine a light on these pigmented cells on the back of these toads, they change their pigment distribution. They're directly light sensitive. And Iggy isolated from those melanophores an opsin photopigment, which he called melanopsin. And then subsequently, Iggy and we and other people all around the world found melanopsin in the ganglion cells of mammals and other vertebrates. But it's caused endless confusion, of course, because people think that melanopsin is like melatonin. And of course, melatonin was called melatonin because it changed the pigment aggregation of pigment cells, the melanophores. So we have 2 names from different directions associated with the pigment cells, the melanophores of frogs, melatonin and melanopsin.
And I've alluded to this already, Russell, but in your Sunday Times bestseller, your latest book, Lifetime, available at all good bookstores, I'm sure, you describe how sleep can have such a profound effect on mental and physical well-being, implications for a range of systemic diseases. But I think not just individuals, but do government have a fairly blasé approach to the importance of sleep? And if you think about government campaigns about stop smoking, eating one of your 5 a day, is it as important as that people should be respecting their sleep cycles and so on?
Yeah, I think it is, Ian. And it's really fascinating because we've talked about this sort of strand of a fundamental understanding how light is regulating the circadian system of animal models and humans and things. But I got into human research and the importance of sleep because of a casual conversation in a lift in Charing Cross Hospital. And I was talking to a psychiatrist and he said, oh, you work on sleep, don't you? And I said, well, sort of. He said, well, my patients with schizophrenia have terrible sleep. And that's because they don't have a job. So they go to bed late, get up late, miss my clinic and don't have friends. And I thought that was not a sensible way to think about the sleep-wake problems of individuals with schizophrenia. So we actually started, and Katharina Wolf, one of my colleagues, we looked systematically at the sleep-wake cycles of individuals with a diagnosis of schizophrenia. And they weren't just bad, they were smashed. I mean, unbelievably bad. And that led us to sort of think about the impact of circadian rhythms and sleep on mental health and other health conditions. And so that's, this is, where are we now? We're in the late 90s, where we started to work on humans and human health and sleep and circadian rhythm disruption. And like many people, very interesting, a lot of circadian researchers like myself have also moved into sleep research. And it'll be bizarre when you say it, but most circadian researchers didn't talk to sleep researchers, and sleep researchers didn't talk to... We went to separate meetings, and our meetings in the early days were very comparative. So there were those people working on pigeons and mice and reptiles and plants. And of course, the sleep people were working on humans using electroencephalography to record the different electrical activity from the surface of the skull, the gross changes in electrical activity from the brain. And we as disciplines really didn't talk to each other, but we do now. And in fact, when I established the Sleep and Circadian Neuroscience Institute, we combine sleep and circadian rhythms in the title, but also we came up with a shorthand of sleep and circadian rhythm disruption, SCARD, because it's so often impossible to disentangle what's as a result of sleep disruption versus circadian rhythm disruption, because the two systems are so inextricably mixed. So yes, what sleep and wake is the states of consciousness and sleep, which involve a complete realignment of all the key neurotransmitter systems within the brain. But that timing of the sleep and consciousness is underpinned primarily by a circadian clock. Now is the appropriate time to be awake, and now is the appropriate time to be asleep, but also what's called the homeostatic drive, or sleep pressure. The longer you've been awake, the greater the need for sleep, and that will interact with the circadian clock. So these sort of yin and yang of circadian and sleep pressure interact to drive sleep timing. So it's an incredibly interesting and complicated system. And of course, the quality of our wake state will influence the quality of our sleep state. And as we're all aware, the quality of our sleep will influence the quality of our consciousness. So I think the most sensible way to think about these systems is as a complex interaction between the two. Hence the description SCARD. And what's I suppose I find fascinating is that we're all aware, of what the short-term impacts of SCARD are. So, we've thrown an all-nighter, for example, or we've had shortened sleep for several days. And you see very early on that our emotional responses fall apart. So fluctuations in mood, irritability, anxiety, loss of empathy, the failure to pick up those social signals from each other. Risk-taking and impulsivity. The tired brain does stupid and unreflective things. The fascinating thing is the tired brain remembers negative experiences, but forgets the positive ones. So one's whole worldview when you're tired is biased towards your decision-making is based upon negative experiences. And of course, our cognition can fall apart pretty quickly. Our ability to come up with novel solutions to complex problems goes. Our ability to multitask. So we're all exposed to masses of information coming in. And what we've got to do on a second-to-second basis almost is define what's important and ignore the less important stuff. And our ability to multitask is very much impaired if we haven't had the sleep that we need. Memory consolidation, communication, decision-making, productivity. These are all things that we're aware of when you get sleep and circadian rhythm disruption. If we look at night shift workers, where you can have night shift for 5, 10, 15, 20 years, you see there's a build-up of health problems. And I should just back off. And if you think about it, what our biology needs is the right stuff at the right concentration, delivered to the right tissues and organs at the right time of day. And if you're not getting that, then the whole system begins to fall apart. And so you see increased levels of cardiovascular disease, altered stress responses. And actually, it's abnormal stress responses that may be underpinning many of the problems. Stress gets a bad rap. stress short term is brilliant. It's long term where you have the problems. And so, as I said, activate the stress axis, you're throwing more glucose into the circulation, you're increasing cardiovascular output. And in a sustained way, this is going to increase one's chances for metabolic abnormalities, type 2 diabetes, high blood pressure, coronary heart disease. But also what's fascinating is that high levels of stress, cortisol, for example, will suppress the immune system. And so this may well be the underpinning for the high rates of cancer you're getting on long-term night shift workers. So, for example, very important studies on night shift nurses have shown higher rates of colorectal cancer, endometrial and breast cancer. And the data are so marked that the World Health Organization has now classified night shift work as a probable carcinogen, which is pretty extraordinary. So driving one's biology outside of its normal range for a long period of time can be really, really difficult. And again, going back to mental health, we do know that sleep and circadian rhythm disruption can exacerbate the slide into a mental health crisis. We've actually done some work with a colleague, Dan Freeman, who's in psychiatry, with young people who were showing very poor sleep. They were showing scarred, but also paranoia and hallucinatory experiences. So they were sliding into that psychotic spectrum. And when you partialize the sleep in those individuals, you reduce the level of hallucinatory experiences and the paranoia, showing a really important role that sleep stabilization can play in mental health as a therapeutic intervention. And psychiatry is now really taking, I think, that work on board very seriously. It's been ignored. It's always been dismissed as, poor sleep, it's just an inevitable consequence of the condition. It partly is. Or it's been dismissed on the drugs that people are on. But actually, you can partially stabilize sleep-wake in those individuals, and it can make a difference. It can reduce the slide into a mental health crisis. And of course, we're going back to the drugs we've been working on to stabilize sleep-wake. It's not just the blind they could be useful for, but stabilization of sleep in psychiatric illness, neurodevelopmental conditions, and indeed neurodegenerative conditions. So one of the great problems in Alzheimer's and dementia is the sleep-wake timing begins to fall apart. And individuals are waking up in the middle of the night and disturbing the whole family. If one could stabilize sleep-wake in those individuals, you'd delay the time when they may need to go to institutional care, which would be good for absolutely everybody.
And with the implications of shift work, do you think those warnings are clear enough for those about to embark upon those roles? Or do you think people that are exposed to shift work have the right to make claims against industry in the same way as people that are exposed to asbestos and those sorts of things?
I think it's an incredibly important point, Ian. And I was talking to the industrial sector just last week, saying that, the 24/7 society is here to stay. It's a genie that's out of its bottle. So we can't say stop night shift work. Our frontline staff in hospitals, ambulance drivers, firemen, policemen, we're going to need those individuals. So it's simply naive to say don't do it. But there are things that we can do now to mitigate some of the effects. And I think this is the message that I'm very keen to get out and isn't currently getting out, which is why I wrote the book, incidentally, or part of the reason I wrote the book, because there's stuff that we can do. So if we just think of what an employer can do for employees, well, microsleeps, falling asleep after the night shift. A study showed that 57% of junior doctors on the drive home after the night shift had either had a crash or a near miss. So why aren't we telling those individuals to download an app onto your smartphone, clip it onto the dashboard so it can detect eye movement or lateral movement of the car? And of course, high-end cars have got this technology built in, and alert people that they're falling asleep at the wheel. And that's just easy stuff we can do now. Poor physical and mental health. Well, in people who have shortened opportunity to sleep or are night shift workers. So why aren't we introducing higher frequency health checks to catch the diabetes, the chronic heart problems before they become chronic, so that medications and interventions can take place? obesity, type 2 diabetes, metabolic abnormalities. What type of food are we providing our night shift workers? It's about as bad as it can get. High fat, high sugar, vending machines full of chocolate bars and sugar-rich bars. And we have a really robust and important food industry in the UK. I don't understand why nobody has come up with tasty, protein-rich snacks, easy to digest, that the night shift community can sustain themselves overnight. I know it's tricky, but as you say, the education isn't there about why one should be careful about one's diet on the night shift. There's lots of anxiety about sleep apps, and they're broadly speaking a waste of time. They are profoundly misleading in lots of ways, and people can get very anxious about being told, you got a good night's sleep or a bad night's sleep, or you need to do this and that. They're okay for a bit of fun, but don't take them too seriously. I think you have in some sectors, the divorce rate, for example, can be some 6 times higher for night shift workers compared to day shift workers. We should not only provide education about the potential harm that night shift can do to the workforce, but also the people they spend their lives with. And a realization from a partner that this person hasn't turned into a monster. This is the inevitable consequence of somebody who's driving their biology outside of their normal range. One of the people at the industrial meeting last week was telling me they were conveying all of this knowledge to a woman, and she started to cry. And she said, I feel so guilty. I feel so terrible because of the way I've been treating my husband, who's a night shift worker. And I just had no idea of the emotional, the cognitive, and the physiological issues that he's having to put up with. So education is so important. The other thing that I think is really worth trying, and we don't have the data for it, is that we know that the problems that you get build up over time. So should we limit night shift work to, let's say, 4 or 5 years, stop, and then you wait 4 to 5 years, and then you can go back on again? Will it reverse some of the health issues that you see? Now, we don't know whether that's the case or not. And also, one would have to deal with the financial side of it. People don't want to do night shift work usually, but they get paid time and a half, and the money is very, very important. How do we jig it so that people, if they cycle out of night shift work, they still recompense financially? I don't know the answer to that. So I think there's a huge educational piece, and I think there's the great danger that unless employers, employees, and the broader scientific and medical communities don't work together, it's as you suggest, you're going to get litigation, and then the whole debate becomes polarized, and people stop working together to try and find a collective solution to some of these problems.
You mentioned that at conferences there was a big divide between the sleep scientists and the circadian neuroscientists. You never spoke to each other. We have a whole field of myopia management, which has kind of been an emerging topic in optometry over the past sort of 10, 20 years, or more actually. So are circadian neuroscientists talking to the myopia management community at all?
Well, it's interesting you should say that. Those of us who work with light have become fascinated with the whole myopia story because of the correlations between daytime light exposure or bright light exposure and the incidence of myopia. And the correlations are very clear. I heard a speaker the other day showing high levels of vitamin D as a metric of light exposure were correlated with lower levels of myopia. And in the same way, individuals, not a great statistic, with skin cancer, high levels of skin cancer, had lower levels of myopia. So vitamin D and skin cancer being used as metrics for light exposure. And I think there are clear correlations. And so, of course, now in Southeast Asia, I think the myopia incidence is as high as 80 or 90% in some urban populations, less so in rural populations. So something really important is going on. What are the mechanisms? Not really clear. So dopamine release, light-induced dopamine release within the eye to prevent axial elongation of the eye has been suggested as a mechanism. But which of the photoreceptors regulating dopamine is not clear. Some work from Bristol suggested it was the rods, but others aren't necessarily in agreement with that. And how and where that dopamine is being released and how it's getting to sort of the microtubular architecture of the sclera to change its shape is also unclear. Is there a circadian component to it? Well, interestingly, there could well be, because dopamine is regarded as the day signal in the eye, and melatonin, because the eye is producing melatonin at night, is the night signal. And so there could be some disruption of the light-dark dopamine melatonin cycle that might contribute to myopia, but we don't know. But I think it's an absolutely fascinating question. If you think about, we know so little for a condition that's costing worldwide billions and billions and having an irreversible impact upon the lives of very, very many people. And I know in this talk that I heard, there was a great tension because this Australian was saying, what do you tell your kids? Go outside and get some sunlight, but increase your risk to skin cancer. So, obviously, if you're going outside, you've got to get that sunscreen on to reduce the chances of skin cancer. Yeah.
I was thinking about how as optometrists we're starting to measure retinal ganglion cells and ganglion cell loss in our glaucoma patients or glaucoma detection.
Working with Susie Downs in the Oxford Eye Hospital, we've been trying to identify those eye pathologies that may be linked to a sleep-wake timing problem. And one of the things that we assumed that we would find is with glaucoma, there would be poorer sleep because you'd find, less regulation of the clock and therefore the sleep-wake cycle. But we didn't find that at all. And I think it's because our definitions of mild, moderate and severe glaucoma are actually very, very mild. If you think about severe glaucoma in rural China, you have massive blindness. But in the UK and other European and North American nations, glaucoma is caught really early. And so what we would think of as severe glaucoma is actually relatively mild compared to the rest of the world. So I think these photosensitive retinal ganglion cells can take quite a hit before you start to see problems arising. And the key thing, though, is that if you are visually blind and you've lost your rods and cones, but you're still showing a pupil constriction, then you should be encouraged to get out and experience sufficient daytime light to allow you to set the clock to the external world. So visual blindness does not necessarily mean total blindness to the vision community. And you should get the advice accordingly. So, I think it's always important to check that pupil constriction, even though people are reporting no conscious light detection.
I think that point, actually, it's only just occurred to me, but I think patients would just know that their eyes aren't as useless as perhaps they think they are. If you're saying to them, giving them that positive message that maybe although you're sight blind, you're not time blind as a function of. I think it's a really nice thing, and it hadn't occurred to me until now. Really,
Yeah, I think that is such an important point. And of course, it wasn't embedded within the training of most optometrists and ophthalmologists because these cells weren't just on people's radar until relatively recently. And I remember going to a congress and speaking about these, must have been about 15 years ago. And there was a tendency then to say, look, you're completely blind. Your eyes are therefore no use to you. They could be a source of infection because you'll damage them, you gash them, you can't see to look after them. The simplest solution is we take them out, we put a prosthetic in, and then it's easier for everybody. But unwittingly, you may then lose the ability to regulate your clock to the light-dark cycle. You've made people time blind where they were not before. So we've got to raise the profile of the importance of these cells and under those rare, but nevertheless, very, very important conditions.
Yeah, I had a very poor night's sleep recently, a few weeks ago, and that led me to behave impulsively. And I submitted an application to do the Masters in Sleep Medicine, actually.
Hey, well done.
So I'm and the application, the research proposal is actually looking at those gaps in optometric education, and is there a role for optometrists, and on undergraduate syllabus. I don't recall it.
The really interesting thing is that Dennis Dacey, when he did his work on the macaque, he identified the melanopsin expressing ganglion cells because they autofluoresce. So you can actually see them. That's how he did the work he did, because he didn't have the advantage of working on a mouse like we had, where we could, genetically engineer or put reporter genes and stuff in. It's always struck me as being a very interesting way forward.
So, I have autofluorescence on the Optomat instrument in practice. So is that something that could be then measured, but then, we're going through the optical media of cataract sometimes?
Yeah, it's basically what Dacey was able to show. He showed that these ganglion cells beautifully autofluoresce. And so if you see a network of ganglion cells fluorescing, they are the melanopsin ganglion cells, which I think is absolutely fascinating. That observation is not clear in the original science paper. It was a science paper, I think. You have to sort of really read in the methodology to work out how he actually found those cells.
And has anyone looked into whether cataract surgery is actually a cure for SCRD? Are there any actual robust studies?
Yes, what we showed was that following cataract surgery, there was an improvement in sleep. So using the Pittsburgh Sleep Quality Index. And it was sustained for the duration that we studied. And there was no difference between, as I say, UV blocking and blue blocking lenses. My only worry about that is that what we didn't do was measure light exposure of those individuals. So we showed that cataract surgery improved sleep. Was it because those individuals, their vision improved and therefore they got out more and got more light exposure? So that's always slightly made me uneasy about those data. And with hindsight, we could have perhaps done it better. And I'm not sure that anybody actually has addressed that yet. I suspect it's because more light is getting through and, you're not seeing it through a semi-opaque and scattered medium. But I do slightly worry about that issue.
Russell, I think perhaps just a quick question to finish on, if that's okay. Each year we spring forward to BST in March and then fall back to GMT in October. Is that a good thing? Does it take us long to adjust to that change?
It's really interesting you should ask me that, because a couple of weeks ago I was asked by some representatives of the Kazakhstan government who have a single time zone of +5 across 4 time zones. So, you've got in the east, sunrise is almost 3 hours earlier than in the far west. And what are the implications of that? Now, that's an extreme example. And I said my position would be noon clock time should be as close to when the sun is at its highest point in the sky. And we should try and stick as closely to that as possible. But clearly it's tricky. And so we invented time zones. But springing forward and falling back in the spring and the autumn, I think is a bad idea and has actually been associated with, so when we lose an hour, there's a blip in the accident rate, there's a blip in people showing depression, there's a blip in cardiovascular problems. Now, I think the issue is that if we are already sleep deprived because we're not getting enough sleep as a society, taking yet another hour away makes things worse and stresses us more, and therefore you unmask some of these conditions. But I would be in favour of no daylight saving time. I know I really irritate my Scottish friends who want an extra hour on the golf course when the nights need to be a bit longer for them, but I have to say I'm not desperately sympathetic about that.
But if we settled on BST and just decided we're going to stay there, would that present more of an issue than just settling on GMT and sticking with that?
Well, as I say, I think we should define noon as when the sun is at its highest point in the sky. And so that's the criteria for me. And, some studies by Till Runneberg, for example, have shown very nicely that if you look at the single time zone that spans the continent of Europe, from eastern Poland to western Spain, if you look at when people get up and go to bed, as you go across the continent, it's delayed as you get into Spain. Now, there's some cultural issues there as well. But essentially, people are getting later and later and later, following the pattern of the sunrise as you go across the continent. There's also been some fascinating studies from the States showing that people living in the western portion of a time zone have poorer health and lower productivity, which is really interesting. And of course, that's the sort of thing that you're getting after a shift to daylight saving time. So my feeling is that we stick with clock noon being as close to astronomical noon as possible.
Russell, it's been fascinating talking with you, and I think we could keep going for another hour at least.
Well, thank you. I've really enjoyed it, chatting to you.